Yes, first we will have Jeroen Sonke to present his work. And Jeroen Sonke is a geochemist and CNRS Research Director of Geoscience Environment in Toulouse in France. And Jeroen is actually one of the pioneers working on using mercury stable isotopes to understand the mercury sources, transport, and transformation processes across the full extent of the mercury cycle. However, Jeroen has also interests and worked with other trace metals and microplastics. And Jeroen have received a couple of ERC, European Research Council grants, and is now since 2020 involved in the Marie Curie Innovative Training Network, GMOS, with 15 PhD students exploring environmental mercury. I could talk for a long time on Jeroen, but I think the very best way to present Jeroen is to let him speak and give his very interesting presentation that we will have now. The floor is yours, Jeroen.
Thank you, Runa. Thanks, John. So let me share my screen. So yeah, thanks again to everybody at AMAP for inviting me and Jens also, who's going to speak in the second half. We're both going to be doing 20 or so minutes talking about mercury isotopes, mostly in Arctic biota, as you will see. So the real pioneers were people like Francis Aston, who discovered mercury isotopes in around 1918, 1919, I don't know the exact year. And he's the man who invented the mass spectrograph when he was at Cambridge. And this is the machine, the instrument you can see in his, either in his garage or in his lab. So you will see a sort of a vacuum tube here on the right. There's a big electromagnet and a whole bunch of other stuff to make it work. And every other week, this man would explore and discover isotopes of a different element. And the way his instrument works is he would introduce mercury in the vacuum tube, somehow get it ionized, probably by heating it. And then he had these velocity selector plates, a high voltage to accelerate the ions, and he accelerates them into a magnetic field, and that's how you separate the isotopes. And then after separation, he basically hit the isotope beams on a photographic plate where they would show up as lines, and that's how he identified the isotopes of the elements. And I'm showing this mass spectrograph and the principle of how it works because today the modern equipment we use, the multi-collector ICP-MS, is based on the very same principles. You still have, you generate ions, you send them into a vacuum tube, you have a high voltage to accelerate them into your magnetic field, here on the top left, which separates the different ion beams into what we call Faraday cups. So just the detection and the ionization system is a little bit more fancy. We use a plasma to ionize the ions, the element mercury. And we have these Faraday cups that measure the current of the ions that basically hit a metallic plate. But other than that, it's still the same physical principle of measurement. You might know that mercury has 7 different stable isotopes. You can see the abundance here. One of them, 196, we never measure because it's not very abundant. But the other ones are all fairly abundant and easy to detect, from 198 all the way up to 204. So when you have 6 isotopes to measure, that's going to give you 5 isotope ratios. We all agreed on putting 198 in the denominator of the ratios we measure. And then, similar to oxygen or carbon isotopes, we report our data in delta values, which is the ratio you measure on your sample relative to an international standard. That's NIST 3133. And then it's the variability in isotope ratio and in isotopic abundance is expressed on the per mil scale. And so when you have elements with multiple isotopes like mercury, you measure these different isotope ratios and delta values. And when you plot one against the other, you'll actually get, most of the time, very good relationships. This is an example of delta 202 against delta 200. Because the reference to 198, the mass difference here on X is 4, the mass difference on Y is 2, and that's going to give you a slope of 2/4, which is exactly what we measure when you plot all the natural data. So this is what we call mass-dependent fractionation, and you could very well measure only one of these delta values rather than the other. It gives you exactly the same information. And for most elements in the periodic table, it goes like that. Now, what's special about mercury, and many of you know that, is when you start looking at the odd isotopes, so now we have small delta 199 over 198 against delta 202, you don't get this mass-dependent behavior with these predicted theoretical slopes. Your data is all over the place, and this is what we call mass-independent isotope fractionation, or MIF. In this case, it's odd MIF, and that's what we will mostly discuss today. And what we then do is we actually use a different symbol, a different isotope signature that we call capital delta 199 with this capital Greek delta symbol. And it's basically the difference between the measurement you make, the observation of the small delta 199, for example, here 5 per mil, and the delta 199 you would expect from theory and based on the delta 202. So it's the difference between your observation and that theoretical mass-dependent fractionation line. So these capital delta 199 or cap 199, they can be positive if you have an excess of odd 199 isotopes, but they can also be negative when they plot below the 9. That's when you have a deficit. You don't have as many 199 isotopes as you would expect from mass-dependent fractionation. And so it just generates more signatures that we can play with. And this turns out to be mostly related to photochemistry. That was discovered in a paper by Bridget Bergquist when she was a postdoc with Joel Bloom, paper from 2007 in Science. And it's a very elegant paper because they combine both experiments on photoreduction of mercury and observations on biota. So their experiments were really pretty simple. They mixed up methyl mercury with dissolved organic carbon, humic acids. And a second experiment where they mixed inorganic mercury. They put it on the roof of their lab in the middle of summer in sunshine, and they just bubbled the solution with air. And so what sunlight does, energy gets absorbed by the organic complex, shuttled to the mercury binding site, where mercury is then reduced to mercury 0. So organic matter gives 2 electrons to mercury, becomes mercury 0, and because you're bubbling it with air, the mercury 0 is removed from your reactor. And when they did that, both for methyl and inorganic mercury, they would measure these positive cap 199 values in the residual methyl mercury and inorganic mercury in solution. And they didn't measure that, but other people did. You can measure the negative cap 199s in the mercury 0 that's evading. So you're gonna have to have mass balance, of course, and that's what they saw. That's how they discovered how photochemistry does this. And then, like I said, they analyzed a lot of fish from the Great Lakes, Lake Michigan, and so fish contains mostly methyl mercury, and they found the same positive cap 199s in the methyl mercury tissue all the way up to 5 or 6 per mil. I'm not going to go into the detailed photochemical mechanism. It has to do with nuclear spin. Odd isotopes have nuclear spin, even isotopes don't. These are very complex radical reactions, and that's how the isotopes get sorted, even from odd isotopes. So let's look at some of the applications of what you can do with all these signatures. First one, I just wanted to look at very simple systems from the literature, not necessarily Arctic, but we'll get there. This is from my former PhD student, Laure Lafont. We worked in the Amazon 15 years ago on gold mining. And we were analyzing fish from the Amazon River and human hair from indigenous populations. So throughout the talks today, you will see this diagram, delta 202 on X and cap 199 on Y. So you're mass dependent against your mass independent fractionation. So you'll see that fish in the Amazon River, it has slightly negative delta 202. sort of close to zero, sometimes a little bit positive Cap199, so a little bit of photochemical reduction of methylmercury going on. Photochemical breakdown, essentially, of methylmercury. But pretty homogeneous group of fish data. And then when you measure the human hair of populations that consume that fish, and these are indigenous people, so twice a day they eat fish on average, you'll see that the Cap199 value is exactly the same value of the fish. That signature did not change from basically from diet to biomonitor tissue to the hair. However, the delta 202 shifted is about 1.5 per mil heavier in the hair than it was in the fish. And that when you work on carbon and nitrogen isotopes, that should ring a bell. Nitrogen isotopes tends to do that as well. Here you have a 1 trophic level shift. And mercury isotopes seem to behave like nitrogen isotope in that sense. You get mass-dependent fractionation. Heavier isotopes are enriched in the next trophic level. All right, let's look at some other populations. We analyzed hair from people in the lab in Toulouse. So we plotted over here. And then when we accumulated data on Mediterranean, Atlantic fish and mussel and shrimp and oysters and all that, kind of plotted over here. So again, the Cap199 pretty much stays the same between diet and consumer. Then there was another study from Joel Bloom's group, Sherman et al. 2013. They worked on a dentist population in the US, and then Bloom also he put out some of the first tuna data on the Gulf of Mexico that I plotted up here, and you get the same trend, right? Average of the tuna fish kind of agrees with hair of this American population. So Americans look like tuna, French people look like oysters, and people in the Amazon, and so on. So we're still not hitting the Arctic. The first Arctic study in this context came out from Elsie Sunderland's lab, working on the Faroese Islands with Philippe Grandjean and I think other Scandinavian scientists. So this was the PhD work of Mei-Ling Li. And they were working on pilot whale and on whalers, consuming pilot whale, of course. And I plotted up the pilot whale data here in the purple diamonds. And pilot whale are a piscivorous species. They eat other fish. And you can see that pilot whale are shifted just like humans that consume fish by about 1.5 per mil relative to the sort of this marine North Atlantic, Gulf of Mexico marine fish average. So guess where the whalers are going to end up? Because the pilot whale kind of looks like human populations. Not exactly, but it's kind of shifted in the same direction. So if I add the whalers, you can add another 2 per mil. And it all makes sense, right? You're just seeing trophic magnification here, and about a 1.5 to 2 per mil mass-dependent fractionation step for each trophic level. And again, the Cap199s, they stay perfectly constant because within your body, within your metabolism, there's no photochemistry going on. There's no possibility to change that signature, in particular in the middle and upper trophic food web. So I think that's all pretty interesting, and definitely in the Arctic, there's a lot of opportunity to use this approach to look at human exposure sources and how they might be changing over time as well. Okay, so then I wanted to spend a little bit more time on exploring other applications of mercury isotopes in marine ecology, some in the Arctic, some not. We did quite a bit of work between 2008 and 2015 on the Alaskan Arctic, where we worked together with scientists from NIST. NIST has a very nice specimen bank where they archive biological samples since 3-4 decades ago, many different species. And with David Point, who at the time was working at NIST, he's now in our lab in Toulouse. We started working on this, and we had a PhD student, Jeremy Masbou, who did all of these analyses. We were working on a latitudinal gradient that was also a sea ice gradient, insolation gradient. Many different things change on this gradient. Different species. We started out on birds, Murray eggs, and then we moved into ringed seals, polar bears, beluga whales. For both whales and bird eggs, we really had the full gradient at our disposal. And then all of the data together kind of plots up like this, delta 202 again, Kep 199. So you get that kind of that same variability that we just saw in the biota when you go from the Amazon River to the different marine environments. And actually the sort of the rough trend that you see in here where you get an enrichment in the heavy isotopes as the Kep 199s get larger, that's what happens during photochemistry. When methyl mercury is photochemically broken down, the product, inorganic mercury, is going to be enriched in the light isotopes. That's always the case. When you have a one-step reaction, the product is always enriched in the light isotopes. And the methyl mercury that stays behind, it gets heavier and heavier. And that's what you're seeing here. As you follow that reaction and you get a higher Kep 199, the delta 202 gets more and more positive. It gets more and more enriched in the heavy isotopes. But beyond that, you see for individual species, there's different slopes, there's mixing going on. All of these species consume other species at lower trophic levels. And so there's potentially a lot of information here. So when you get a data set like this, and you think about its interpretation, you're gonna have to be thinking about different factors. External factors like the inorganic and methyl mercury physicochemistry, photochemistry, and those are factors that relate to climate. They can relate to different mercury deposition sources in the biogeochemical mercury cycle, sources that might change, right, from the Gulf of Alaska all the way into the Arctic Ocean. You're going to have to think about metabolism, methyl mercury breakdown within different organisms. You're going to have to think about excretion. So that's a delta 202 shift that we talked about. Jens is going to go into more detail about it. What aspect of metabolism is actually doing that? And you're going to have to think about, what part of that trend is related to food web, to foraging in different areas, according to the season, migration, or ontogenetic shifts at the different life stages of your species. So in two different studies by David and Jeremy Masbu, we hypothesized that the trends we were looking at, so as you go north, here you have latitude on X, these Kep 199 signatures, they basically come down. For the two different species where we have the spread, both for the beluga whales and for the Murray eggs, we see that trend. It's going down as you go north. And we interpreted that at the time as most likely a sea ice effect. The more you have sea ice in your foraging area, the less you have photochemical breakdown to the point where your Kep 199 just stays zero. It doesn't move at all because there's very little photochemical breakdown. So that's the hypothesis we developed in these studies. But you can definitely think about other things. I talked about insulation. It turns out that between 55 and 70 degrees north, there's not that much change in insulation over between those two latitudes. But you can think about different sources of DOC that's involved in the photochemistry. Think about different baselines, right? It's like nitrogen isotopes. The methyl mercury in these marine systems, they might have a different Kep 199 baseline, et cetera, et cetera. So we hypothesized mostly about sea ice, but you can never exclude when you propose that it's something else. And that's something else. That's something that came out in recent work, 2020, by Marina Renedo, a postdoc who worked with Jeroen E and Paco Bustamante. They were now working on different sea bird species, little auk, where they analyzed feathers. The yellow data points here and the orange ones, those are the populations that they worked on. So we're now in the Eastern Arctic, if I say that well. You had our data here in the Alaskan Arctic. So they worked on a gradient here. Same thing, latitude gradient. So in their data plot, again, kind of the same, right? Kep 199 here, variability. The green data is the Alaskan data. You have it going down as you go north. And in the Eastern Arctic, none of that happens. Pretty much stays the same or even goes up a little bit. So why is that, right? And yeah, you have to think about different hypotheses. I mean, is this mostly, are these ice-free waters compared to the Alaskan Arctic? Maybe. Or is it a migration thing that these birds actually, they show that signature, but that's because they're feeding further south in summer where they get most of their mercury. I don't remember what their preferred hypothesis was in there, but this is kind of an open question and might be some other factor, right? Okay. Moving out of the Arctic a little bit to show you other applications of what we can do with these mercury isotope signatures. This was a study, again, with David Point now on sharks in the Pacific, looking at great white sharks, where there were many open questions on what the great whites were feeding on. Were they feeding inshore mostly on pinnipeds, on seals? Were they feeding offshore at the surface, epipelagic fish species, or down in the twilight zone on squid? And so Gaël Le Croisier, the postdoc, he's a shark specialist. He's got his hand on the different prey species, and that's very important. You have all the shark data here in the circles, 2 different tissues, muscle and skin, and then you have the prey populations. Here you have the seal, you have the epipelagic fish, and in green here you have the squid from the twilight zone. And it's clear if you look at the signatures that they were not feeding so much on epipelagic fish, but more on squid and on pinnipeds. And apparently that was fairly new to the ecologists working on these shark species. Okay, so you can understand foraging patterns. And of course, in the future, you can understand how this might shift with changes in ecology according to biodiversity, overfishing, climate change issues. Another application kind of started with the work by Jeroen Bloom, who worked on fish species again in the Pacific Ocean. And he analyzed different species as a function of depth. So you have the delta 202 and the Cap 199 signature all the way down to 600 meters. And you can see that both decrease as a function of depth. For Cap 199, that's perfectly understandable. You have more photochemistry going on at the surface, more photochemical breakdown of methylmercury, and you get these higher signatures, whereas at depth, you're out of the photic zone. And then the ecologists, they jumped on this to start using the Cap 199 tracer as an indicator of foraging depth. So again, here on the shark, 2 populations from the Pacific and the Atlantic, where you have different oxygen depth of the oxygen minimum zone. So working on blue sharks this time. So you can see that in the Pacific population, average Cap 199 was 1.6. And in the Atlantic population, it was 1.3, a little bit lower because the oxygen minimum zone is lower in the Atlantic Ocean, and the blue sharks were simply feeding at a deeper depth. So that's the hypothesis, right? It makes sense. But I do have to say that would be nice to really uncover the same trend that Jeroen Bloom saw in the Pacific. So we need to look at these variations in Cap 199 in the different ocean basins to really solidify these interpretations. The last example before I hand over to Jens is a recent paper that came out 1 or 2 months ago by Lucien Besnard, another shark. The shark people keep contacting me, and they develop cool applications. So now it's mercury isotope clocks with the Cap 199. They were interested in tracking when, at what age, juvenile sharks in the Pacific, around on the Mexican coast, basically quit their coastal habitat and move offshore to lead their bachelor life. And we know that in the coastal ecosystem, you have higher anomalies than offshore, where they feed on deeper prey. It's the same idea. So here you have a plot with the total length of the juvenile shark that grow into adults, so from 60 to almost 2 meters long. And you can see how the Cap199 for the juveniles in the coastal ecosystem is high, and then for the near adult population, it's low. And then everybody else falls in between. It's noisy data. So what they then used is the turnover time of methyl mercury within the shark body, which apparently is pretty well known. I think it's on the order of a couple months, to develop the Cap199 signature in an isotope clock. And they learned that it takes about 2 years before the sharks move offshore. And then knowing that, they know pretty much the size of the shark before, when they start moving offshore, probably around 1 meter long. And that helps them in improving conservation of these species in the coastal environment where you have a lot of fishing going on. All right, so pretty interesting applications, right, that you can do just with these two simple signatures. There's many more signatures to mercury isotopes, but definitely alone with these two, you can explore a whole bunch of big questions in ecology or in environmental chemistry. So over to Jens.